Thank you for the kind introduction. I'd like to also thank Philippe for his kind invitation so that I have the opportunity to come here and share with you some data from our work in the lab related to characterization of human blood monocyte subsets. So we know that hematopoietic stem cells can differentiate and generate into different types of immune cells, as you can see here. And we also know from here that all these different immune cell lineage has got different type of subsets described for them. Here are some examples listed here for T cells, B cells, NK, as well as dendritic cells. But for monocytes, for a long time, people has believed that monocytes is just a precursor cells for the generation of macrophages and for the generation of dendritic cells. But in fact, it is more well accepted now that monocytes in its own right is also playing a very important role in modulating immune responses. And for monocytes, it's also not surprising that it is not homogeneous in the blood. It is also heterogeneous, and this has been first described by Ziegler-Heblock in 1989, where he used two markers on the surface of monocytes, CD14 and CD16, to broadly divide these monocytes into two subsets. Here I've labeled them, the one that expresses CD16 as 16 positive, while the one that does not express CD16 as a CD16 negative monocytes. And here I've listed some examples of differences that has been described for these two subsets throughout studies that has been done since 1989. It's not meant to be exhaustive, just some brief example of differences between the two subpopulations. And in humans, we know that the 16 positive population makes up of a minor population of the total monocytes of about 20%, while the 16 negative monocytes made up the majority of the bulk of the 80% of total monocytes. And in response to LPS, it has been described that the 16 negative population tend to make more IL-10 and seems to be playing more important role in tissue repair and homeostasis, while the 16 positive subpopulation in response to LPS seems to be making higher levels of TNF-alpha and has been labeled for a long time as the pro-inflammatory monocytes. And so it is actually not surprising to see that this subpopulation has been described to be expanded in during many types of inflammatory conditions, which I will also share with you later. And one interesting aspect of the 16 positive monocytes is this patrolling behavior that has been described more recently. And this patrolling behavior of on the vessel wall of the blood vessels seems to be indicating that this cell type is playing a centennial role and is sensing the microenvironment and getting ready to extravasate into any inflammatory site when there is an infection or when there is inflammation occurring. And here I've also shown you some of the markers that are differentially expressed on the monocytes. In this case, only 3 markers has been depicted here because these 3 markers has been very important to identify the homologous population between human and mouse. For a long time, the homologous population between human and mouse has not been well established due to the lack of markers between the humans and rodents. But now with these 3 markers, the homologous population between mouse and rodent has been much better well defined. And also, CD16 is a marker that is used to segregate the two subpopulations, and this marker is also Fc gamma receptor R3. So one of the stories I'm going to share with you today is basically looking at the role of monocyte subsets in antibody-dependent cellular cytotoxicity. So for Fc gamma receptor, we know that they play important role in antibody-dependent inflammatory responses, and they promote antibody-dependent cellular cytotoxicity, known as ADCC, also play important role in phagocytosis, cytokine release, and oxidative burst. And the two main cell type that is important in performing or making use of Fc gamma receptor for its immune response is NK cells and macrophages. Here, using tumor cells as an example, I've shown you that NK cells can bind to antibody-coated tumor cells and lyse the tumor cells through the release of perforin and granzyme. Whereas for macrophages, it actually kills the tumor cells or antibody-coated tumor cells through a Fc-dependent phagocytic event through the degradation of the tumor cells in the lysosome after it has phagocytosed the tumor cells. So the event of the killing of antibody-coated cells appears to be very different between the two cell types. But what about monocytes? So monocytes has been very loosely attached with macrophages because these two belong to the same lineage, but very little is known about how they perform ADCC, which I'll share with you some of the data that we have gotten here. So besides Fc gamma receptor R3, which is CD16, that is I've shown to be differentially expressed between the two subpopulations, in fact, Fc gamma receptor R1, CD64, okay, is also differentially expressed between the two subpopulations, while Fc gamma receptor R2A, CD32A, expression is actually similar between the two subsets. And this is just an example of some of the Fc gamma receptors that are expressed in humans. So based on that, we would like to find out if monocytes actually can perform ADCC and if there's a differentiability between the subsets. But before that, let's see how an NK cell can typically kill antibody-coated tumor cells. So here I've got a time-lapse images showing how NK cells kill this breast cancer cell line that has been coated with antibody against the antigen HER2 using the antibody Herceptin. And if you focus on this is the tumor cells, and you focus on these NK cells here. So the NK cells interact with the tumor cells, and then after that it kills it. So what about the monocytes? How do they do it? So again, here I have two tumor cell line, two images to show you. On the left is again the breast cancer cell line, which is coated again with Herceptin, the antibody against HER2. And you focus on this monocyte here, interacting with the tumor cells. So it interacted with the tumor cells, and with a bit of struggle, finally it will kill off the tumor cells. And on the right, we have got Raji cells that are coated with rituximab, anti-CD20. And you see a red cell here because we added povidone iodide into the supernatant. And once the cells undergo lysis, they will uptake the dye and turn red. So this is actually a cell that has lysed. So if you focus on this Raji tumor cells and you focus on this monocytes, you will see how the event takes place. So they interact, and very soon after, the cells is lysed and uptake povidone iodide. So it seems like from these images that monocytes perform ADCC in a fashion very similar to NK cells, unlike the macrophages where they phagocytose the tumor cells and then it that degrades the tumor cells in the lysosome. So to further show if there's a differentiability of the monocyte subsets to perform ADCC, we use 3 different tumor cell lines as our model. In this case, we use a lung cancer cell line, Raji, a B lymphoma cell line, and the breast cancer cell line. And the antibody we have used to coat the cells is either chimeric or already in therapeutic use in the clinic. So such as GM2, which is a chimeric antibody for the binding to GM2 on A549, and rituximab for Raji, and Herceptin for the breast cancer cell line. So after isolating the monocyte subsets, we put it together with tumor cells that are not coated with antibody, and we can see here that for both of the subsets, there is hardly, if any, killing of the tumor cells. If there is some slight killing, the killing occurs at a very high effector to target ratio. However, when these tumor cells are coated with the respective antibody, then we see a very different picture. We see a huge increase in the ability of the 16 positive cells to now lyse the antibody-coated cells in all the 3 models. And interestingly, the 16 negative cells, whether it's coated or not coated with the antibody, we do not see any difference in the ability of these cells to kill. So it seems like ADCC is performed exclusively by the 16 positive monocyte subsets. And how does it compare to NK cells in terms of its efficiency in killing? So taking cells from one donor, the NK cells and the two monocyte subsets, and taking them to perform ADCC, we can see that the 16 positive cells are only slightly inferior to the NK cells in its ability to lyse antibody-coated cells, indicating that it is also a pretty professional killer. And this event has to take place in the presence of antigen-specific antibody, because if we add the irrelevant antibody into the ADCC assay, we do not see any enhancement in killing, so indicating an antigen-specific event is necessary. So, so far I've shown you the killing of mainly tumor cell lines, but in order to move a step more forward from bench to the bed, we would like to find out whether these cells, these monocytes, are able to also, able to kill primary cells as well. So to do that, we obtain some primary B lymphoma cells from patients in the clinic. They are suffering from Hodgkin B lymphoma. Here I show you data from 3 patients, 2 of which the tumor cells are obtained from the bone marrow, and patient 3, the lymphoma cells was collected from the peripheral blood. And for all 3 cases, we can see that the moment we coated the cells with rituximab, the anti-CD20 antibody specific for the antigen on these tumor cells, we see enhancement in the killing of these cells by the CD16 positive monocytes. Again, there is no effect if we add the irrelevant antibody into the ADCC assay, indicating that CD16 positive monocytes is also capable of killing primary B lymphoma cells. And we know that NK cells can perform ADCC, and for a long time it has been described that the cocktail of IL-12 and 15 can enhance NK cells to kill tumor cells better. And we can also see this in our own hands. With treating of NK cells with IL-12 and 15, we can enhance its ability to kill antibody-coated cells. But are we able to also enhance the ability of the 16 positive monocytes to also perform ADCC? And if we tried using IL-12 and 15, which is a very specific activator for NK, we do not see any effect in the CD16 positive cells. But however, if we were to use cytokines that can actually activate monocytes, now we see that the CD16 positive monocytes are able to then enhance in its ability to kill tumor cells. But in this case, it has to be in tumor cells that has been coated with antibody. So it's not a very general activator of activating the monocytes, but rather it has to be in the context of antibody coating of the tumor cells. And this has no effect on NK. And we notice that there are other stimulants that can do the same in activating monocytes, the CD16 positive monocytes, to become better killers. For example, here, S100A9 is also capable of enhancing the monocytes to kill, as well as some toll-like receptor agonists such as R848 and LPS. So to summarize, we can see from here that IL-12 and 15 cocktail can activate NK cells to kill, while these other factors has no effect on NK cells, while IL-12 and 15 has no effect on CD16 positive monocytes. These other factors are able to enhance the CD16 positive monocytes to kill tumor cells to various degrees. And this is something very specific to the CD16 positive monocytes, because we do not see any enhancement in killing in the CD16 negative cells, even we were to activate them with these factors. So indicating that ADCC by 16 positive monocytes are selectively enhanced by cell type specific factors. And this is, to me, a very important piece of data, because it could mean that, for example, in the microenvironment where you have a different context, perhaps either NK cells or the CD16 positive monocytes might be activated in different scenarios, leading to different outcomes. So we know that the 16 positive monocytes kill in a fashion similar to NK, and we know we can enhance them to be better killers, but what is the mechanism involved in their killing? So to address that, let me first recap. So we know that NK cells can kill target cells through the release of perforin and granzyme. As for macrophages, it's also well described that they are able to kill tumor cells either through ROS, NOS, or TNF mediated events, either singly or in combinations. So for monocytes, since it's a myeloid cell line, we've decided to address whether it kills the tumor cells through one of these three mechanisms. So to address ROS and NOS, we have used specific inhibitors that inhibit ROS as well as inhibit NOS. So we can see from here that the addition of ROS or NOS inhibitor do not inhibit the CD16 positive monocytes from killing its target cells, indicating that these two pathways is not involved in CD16 positive monocytes performing ADCC. But what about TNF-alpha? So using a combination of anti-TNF-alpha and anti-TNF receptor, we can now completely block the ability of these monocyte subsets to perform ADCC, indicating that ADCC by 16 positive monocytes is mediated through TNF. And we also know that factors that is added to the monocytes can actually induce the expression of CD16 on monocytes. So we would like to also ask, if we were to induce the expression of CD16, can we now make the CD16 negative population to become killers as well? So in the next slide, I've shown you that if we treat CD16 negative monocytes with these factors overnight, either MCSF, TGF-beta or IL-10, we can induce the expression of CD16, with IL-10 showing the most potent effect in upregulating the CD16 expression. And now do these cells has the ability to kill now that they have upregulated their CD16? And indeed, yes. They are able to kill antibody-coded cells now, and it looks like there is a linear proportion between the expression of CD16 and their ability to kill, as we can see here. And using IL-10 as an example, we can also see that the blocking of the addition of blocking antibody against anti-TNF-alpha and TNF-alpha receptor can also inhibit these cells from now killing its target cells, again showing that the killing is through a TNF receptor mediated event. So to summarize, I've shown you so far in humans that the CD16 positive monocytes is able to exhibit ADCC, and this is mediated through a TNF-dependent mechanism. And I also show you that in vitro, they have the ability to kill antibody-coded primary B lymphoma cells. And these cells' ability to kill can be further enhanced either by the addition of interferon gamma, S100A9, or toll-like receptor agonists such as R848 or LPS. And this has, in my opinion, a therapeutic application, especially pertaining to the context of antibody therapy for cancer. So something that we should also look into, apart from the studies that has been done on NK cells. And this is this report here by a group reporting about mouse model at the end of last year in Immunity actually was a nice timing, showing that even in the mouse model in vivo, they showed that this population of monocyte subsets, Ly6C low, Fc gamma R4 positive, which is the homologous population of the CD16 positive monocytes, in fact, in vivo, is the subset that is specifically performing ADCC on antibody-coated B cells, mirror very nicely what we have or have described in human settings. So again, indicating the importance of now looking into this subset as well in the setting of tumor treatment. So in the last 5 minutes, I would like to quickly share with you some new nomenclatures that has been described for the monocyte subsets. So far I've told you about monocytes being broadly described into, broadly classified into two populations. But in fact, in 2010, the nomenclature committee has decided that, or proposed that monocyte subsets should now be divided into three, based on the gating as shown here. And so there are three subsets instead of two now, and the subdivision of the three subsets is due to the division of the 16 positive into two subpopulations, as you can see here, resulting in the generation of a new intermediary transitional subpopulation. And the committee also proposed that for consensus reason, that the three subsets should now be labeled as classical, intermediate, and non-classical. So in order to better understand what these three subpopulation is doing at steady state or in homeostasis, we isolated the cells and performed gene expression array on the three subpopulation. And based on principal component analysis or clustering, we can see that the intermediate and the non-classical population cluster closely to each other, while the classical population is a bit further away, indicating that they are more closely related to each other. And looking at the number of differentially expressed genes, we also see that the number of differentially expressed genes between the intermediate and the non-classical, in fact, has the lowest number, again showing that they are more closely related to each other than to the classical monocytes. And if we look at some of the surface markers that are expressed, differentially expressed by the monocyte subsets, we see an interesting continuum in the expression of these surface markers between the subsets based on the gating strategy that we have used here. So we see a continuum either in the increase or decrease in expression of some of these surface markers from classical to non-classical monocyte subpopulation. But what about function? So we also perform some functional analysis. For example, doing a very simple phagocytosis assay with uncoated polystyrene beads, and we can see that if we activate the monocyte subsets with LPS, the intermediate population has the intermediate ability to uptake beads, the polystyrene beads. And in terms of the ADCC that I described earlier on, if we now divide the 16 positive cells into the intermediate and the non-classical, we can see that the intermediate again has a significantly less capability of lysing the tumor cells as compared to the non-classical population. All these pointing to the intermediate monocytes exhibiting functional property at the intermediary level. But however, it doesn't, from all this data, it seems to show that the intermediate population has a transitional property. But in fact, this is not really the case, because from our array, we also notice that there are genes that are most highly expressed specifically in the intermediate, particularly in those genes that are related to MHC class 2 processing and presentation. As shown from the FACS plots here, the highest expression of CD40 and HLA-DR. And also in their response to LPS, this particular population is able to secrete as efficiently as the classical and non-classical in terms of the production of IL-6 and IL-8, while having very poor ability to produce TNF-alpha as well as IL-1 beta. And why do we think that the intermediate monocyte subsets is important or is so interesting to explore? Because in our own hands, looking at patients suffering from SIRS, we notice that these patients tend to have a preferential expansion of the intermediate monocyte population, as shown in this slide, both in the absolute count of all the patients that we have looked at so far, as well as in this FACS plot, as you can see here, the accumulation of the intermediate population. And this is not only just described for SIRS. In fact, doing a thorough analysis of some of the diseases that has been described by others, whereby they showed that 16 positive monocytes are expanded in these range of diseases, looking at those papers more carefully, it pointed mainly to the intermediate population to be expanded. So these indicate that this population is probably playing an important role during disease, but whether it's playing a protective role or a pathogenic role is unknown, which needs further investigation. So based on what we have done so far, characterizing the three monocyte subsets, some of the new information that we do know now about these three populations is summarized here. We know that between the intermediate and the non-classical population, it is a non-classical that is mainly producing the TNF-alpha, and it's also making high amounts of IL-1 beta in response to LPS, while the intermediate is making similar level of IL-6 and IL-8 as the classical and non-classical, while having no capability of producing the other cytokines that we have investigated. And they are also the population that exclusively express CD40 and express highest level of genes related to MHC class 2 presentation and processing. And we also found new markers that are uniquely expressed on the classical, and some markers that are preferentially expressed at a higher level on the classical as well. And we also notice that the classical monocyte in response to LPS, besides making more IL-10, is also making more CCL2, GCSF, and RANTES. So based on that, I would like to conclude my talk by, of course, acknowledging the people who has contributed to the work. I would like to thank Philippe for giving me this opportunity to manage a small group, which you can see here. The work done on the ADCC is mainly performed by the research officer, Wei Shen. She is a very dedicated research officer. And the characterization of the monocyte subset is mainly the work done by the postdoc, Kok Lun, together with Wei Shen and June, which is the other officer. They did most of the facts analysis characterization of the subsets. And I also want to further acknowledge my collaborators. I'd like to thank the microarray core team for their assistance with the gene expression experiments, the flow cytometry core team for helping us to sort out the monocyte subsets, and I would like to thank our collaborators from the Bioinformatics Institution for the analysis of the gene array data. And from the hospital, the two clinicians, Ye-Ching Lin for the B-cell lymphoma, for us to perform the ADCC on the primary tumor cells, and Dr. Zhenwen Chong for his collaboration on the CS project that is still ongoing. Kiowa Kirin provided us with the anti-GM2 antibody, and fundings come from A-STAR, and of course this study would have been impossible without the blood source from the two blood banks in Singapore, which is listed here. So thank you very much.